Thank you, Marco, for that brilliant introduction. First of all, I'd like to thank the organizers and the Center for Personalized Medicine in Oxford for giving me the opportunity to speak here. I'll talk about paradigm changes, and I think that alludes to Professor Novelli has just mentioned. When I was in the dining hall last night here at RSM, I saw this beautiful microscope. This is a Carl Zeiss microscope, and it has a serial number, so I had the chance to look this up in the online archives. And this was sold in London here by Carl Zeiss in 1911. So that's only 60 years after Virchow first formulated his cellular pathology work, which has been seminal. And since then, the microscopes have gotten a lot fancier, and we've gotten a lot better at reading these slides. But really, the way we generate this information hasn't changed very much. We're still at an analog information level in most parts of the world. And I like to raise the similarity to a hand-drawn map. If you look at a slide, that slide is unique. Only a few people can look at it at any one time. But it contains all the information that you would like to get from that slide to guide you where you want to go. But it's limited. So right now, the transition to digital pathology is a complete paradigm change. It's a game changer. It's turning these slides into fully digital, searchable, and structured databases that we can then interrogate using bioinformatics methods and artificial intelligence. And if you want to extend that analogy, we're going from this hand-drawn map to a Google map that could be searched and is interactive in one technology step. And that is really changing the way we're doing pathology since the last 160 years. So let's look at the increased information content. Any of these slides are usually pyramidal images containing all the information at different magnification levels. So you see what depth of information there really is here. But interestingly, we're also beginning to associate this with structured metadata that we can include in these image formats, probably most likely in the DICOM format in the future. And then this is another very interesting point. If you look at ImageNet, this is something that computer vision scientists have used over the past decade or so to train object recognition algorithms. And this is something that you will use on a daily basis. If you do a Google picture search and you say, I want a picture of a colorectal cancer, there's an algorithm running in the background comparing all the outputs and looking for that specific pattern of information. So all of this ImageNet, that's 14 million images, contains 2.8 trillion pixels. The same amount of information is on only 474 pathology slides that are digitized. And that's about the amount of information that an NHS pathologist will read in about 1 to 2 days. So you're seeing that increase in information content. And we're beginning to really mine this data to increase our understanding of cancer biology and how that could impact clinical practice. So what are the key methods? I know there's some pathologists in the room, but I'd like to really quickly allude to the key methods that have shaped this field. As in many other areas where we're talking about image analysis, so that is radiology, facial recognition, etc., deep learning has really been transformational. So deep learning, in particular deep neural networks, are a way to use statistical learning methods that have been modeled after the human brain, essentially, to extract abstract patterns from large image data. And these abstract patterns can then be correlated with specific genotypes, for example, or clinical outcomes. So this is one way in which we can use this image data and interrogate it. Oftentimes these are unbiased approaches. So you're really asking which type of pattern associates with a certain outcome of interest. The other way we can approach this problem is to use our prior knowledge that we've generated by looking at these slides over years and years, and to craft AI-based approaches that will pull out a certain type of feature, so be it stroma content, immune response, cellular shape, that we can then specifically interrogate with image features and outcomes of interest. And I'll show 2 different examples of each of these applications in colorectal cancer. So when you look at most of these studies, this is the way they're set up. You usually need a very well-structured clinical and ideally molecular data set, as well as pathology images. And now a part of this data set will be used as a training set to derive the AI-based classifiers. But there's a problem here. If your training set doesn't include all the diversity that you would expect on population-wide data sets, that is going to be overfit to your training data, so it won't generalize. The second problem is if your training data set is too small, too specific, your algorithm that you're training will never reach any acceptable performance on that data set. So having the right data and structured data is very essential. Secondly, to make sure that what you derive actually generalizes, you need validation data sets and test data sets. Another important point for people not involved in image analysis or digital pathology is that when we're speaking about image classification, we're usually deriving this prediction on a tile level. So all these big slides are split into tiny little tiles that are about 200 by 250 by 250 microns, and the prediction is made on each of these tiles. And usually, the summary prediction for an entire slide then allows to make a classification. So, is everything solved? I think that question's been asked before with the advent of next generation sequencing and genomics, and it didn't really solve everything then. But if you look at the hype right now in the media, it looks like where doctors are out of business, and this isn't just pathology. It's it's radiology, it's ophthalmology, etc., anything where there is an image. Is that true? I don't think so. At the moment, where the field is going is that we're building purpose-built intelligent solutions that are very good at a given task. But really, what we're doing is we're not at the point of a strong artificial intelligence yet. What we can achieve is an augmentation of human intelligence. And I think that's an important point to keep in mind when looking at paradigm changes and how they will impact the future of our area. So, Virco, it's really been a long time where we've used the morphological information for classification, staging, and grading of malignant disease. But at the moment, we haven't really connected this to the precise genotypes that are driving precision medicine or personalized treatment. And the talk today, I want to really focus on how we can use AI algorithms to integrate these two complex information data sets and how we can use this to generate benefit for patient stratification and treatment. And I'm coming back to a similar image from Professor Mon has just shown. And I'd like to highlight one point. So far, we've been looking at morphological phenotypes as pathologists, and we've been providing this information to molecular analysis, but it's been a very, very one-directional affair. We've looked at slides and given information of how much tumor is on a slide to then correct copy number calls, for example, or we've given rough estimates of which tumors would need to go on for molecular testing, as in case of MSI. But now, with AI, we can close the loop. We can actually ask which specific changes on the slide are dictated by the molecular phenotype, and we can achieve a better stratification by iterative connection of molecular and morphological phenotypes. And I think this is really going to be a very, very interesting area where we'll see a number of applications, both in clinical practice in the future, but at the moment also a lot of new information that will empower us to address new research questions. So let me give you an example of how we can use AI-guided tumor annotation or tumor segmentation to improve downstream molecular analysis. And this is an example of using prior knowledge and using AI-based methodology to better extract information from a given histology slide, and to do that in a more reproducible way than we were able to using the gold standard pathology method. And why is that important? It's important because we know that what we do as pathologists, so the annotation of the tumor region on the slide, is very important for the success of molecular profiling. Firstly, we know that tumor cellularity is strongly associated with DNA concentration. We know that our ability to detect any somatic variants in a cancer sample will drive the treatment of that patient. Also, looking at large clinical trials, low tumor cellularity is a common reason for failed downstream molecular testing. The retesting of inadequate samples generates significant costs and sometimes requires a rebiopsy, which causes patient morbidity and delay of treatment. And one thing cancer patients don't have is time. So when we look at the gold standard and look at large patient cohorts that have been annotated by pathologists, you very quickly see that a key problem is the inter-observer variability when asking the question, how much tumor is on a given slide? Right, so this is slides from the ESCOT data set. where we've asked two pathologists to annotate these colorectal cancer sections, and both of these pathologists are correct, but there is some variance how the content of tumor is interpreted, and of course there is then a secondary guess that is made of how many of the cells that you see here are actually tumor cells, and how much of it is stroma or immune cell infiltrate. And so if you look at a given data set, the predictions that two pathologists make, and these are expert pathologists with lots of experience, are not very reproducible. And this then leads to a rather imprecise correlation with downstream metrics, such as DNA and RNA yield. You can see that here across these pathologists, across the cohort of 410 colorectal cancer patients that we looked at here. Now, how does this compare to other gold standard methods? This is the estimate bioinformatic way of predicting tumor purity. This uses single cell gene set enrichment analysis to predict tumor purity from expression data by focusing on stromal and immune signatures. And you can see that the estimate method is quite accurate when you compare it to the gold standard. So this is the absolute method using copy number alterations. But if you compare the estimate predictions across TCGA, and this is the entire 11,000 odd cases of different tumors in TCGA against pathologist scores, you get a big cloud. So there is very little correlation here between bioinformatic prediction and pathologist scores. So what can we do to improve this? This is work done within ESCORT. I'm very grateful to have had the chance to be involved from a pathology side. We've used the 410 cases from the FOCUS randomized controlled clinical trial, patients that were treated by 5FU and or oxaliplatin, with full clinical pathological and outcome data. So the beauty of this data set is that we have a first H&E here that we can use for AI-guided image segmentation. We then have the sequential section that was used for RNA expression profiling on a high-density RNA FFPE expression array. This is the RMAC-XL array. We then have another H&E, which we can again use to run our image analysis algorithm, and then correlate this with the mutation profiles that we derived by a targeted NGS panel, including the 80 key CRC driver genes, as one of the methylation data. So we've got two levels here of information where we can look at the impact of the tissue segmentation on the H&E against molecular data. So what we used to derive this was a pathologist-trained tissue segmentation approach by supervised machine learning using a VGG implemented in the Halo AI platform. So the VGG is the deep neural network, which was originally developed in Oxford. And you can see here a colorectal cancer case, and you can make a guess for yourself how much tumor you think is on that slide, and I'll give you the solution a little bit later on. And now using these trained deep neural networks, we can very precisely segment this case into the different tissue components, so tumor, stroma, mucus, necrosis, inflammation, but also background classes and artifacts such as folds here that you can exclude, and of course glass areas. So you get a very, very precise measurement of all of these regions. And now this allows us, if you look at the detail of this slide, to derive the composition of that tumor down to the square micron resolution. You get very, very precise measures of tumor area across each case within an entire clinical trial. So we don't want to stop here because we know that there's different densities of cells in different tissue types. So we ran cell segmentation within each of these AI-based tissue areas, which then gives you the precise cell counts within each area. And using this, we can now get a very, very precise metric of how much tumor is on that slide. So it's about 538,000 cancer cells, and we also get secondary metrics that are interesting to correlate with biology, such as nuclear optical density, which can correlate with DNA content, or also cell shape, nucleus area, perimeter, etc. Of course, we can run the same for the stroma. We get the precise number of stromal cells, and then we can look at the actual correlation with pathologist estimates. And if you've made a guess, this is the correct number based on area, and this is the correct number based on cell counts. You can see that the pathologist estimates range quite widely. So why is that? So if you now look at the densities, you see something that might be expected, the tumor cell density is much, much higher than the stromal cell density. So we have a systematic underestimation if we just look at the area. And it's very difficult for a human to integrate a measure of density in their assessment of cancer on a slide, because there's also quite a bit of variance there. And AI-based methods can allow us to get more accurate data out of these slides. How reproducible is this method? So this is between the first H&E and the second H&E that I've shown you, with several sections in between. You see that even the area-based measurements as well as the cell-based measurements are very reproducible across this entire cohort. Now, the interesting thing is if you look at this and ask yourself, now how does this impact the downstream molecular analysis? So as I've shown you, we usually underestimate purity if we have a visual assessment of the slide against the deep neural network. So as you can see here, there's a shift of tumor cell content towards higher tumor cell content if you use DNN-based metrics versus visual. And this then leads to an underestimation of the impact of CNAs. The same is true if you use RNA-derived metrics or methylation-derived metrics. So now here, the bioinformatic prediction uses only stromal and immune cell signatures, as I've shown you, to make an estimate of tumor cell content. But there's a lot more on a slide than just stromal, so it's just fibroblasts and immune cells. There's also normal mucosa, there's muscle, there's other components of the microenvironment that are neglected. So using these methods, oftentimes purity can be overestimated. And if you now look at the copy number calls of the downstream molecular analysis, you see that this leads to a correction of copy number calls in about 10% of cases. So we have consistent overcalls by 10% with visual pathology. We have consistent undercalls by about 10% with deconvolution methods. So using this DNN approach, we can get more better and more precise data from the standard H&E section to inform molecular pathology. But it's not only that, you can also, in a very coarse way, of course, look at the precise composition of each tumor between these different components, so epithelium, but also different types of stroma. So desmoplastic, fibroblast-rich stroma with very few immune cells versus stroma that is inflamed. And you can start to look at this across cohorts. And this is a small set of 58 PT1 rectal cancers that were analyzed in collaboration with Helen Jones and the leading group. And you can see this very, very precise metrics here. But of course, you can now also use these tumor-derived metrics to correlate this with outcome. And it's quite interesting that in these untreated rectal cancers, stromal content is already a measure of tumor aggressiveness. So those patients that had a higher stromal cell content had a significantly higher rate of recurrence. And I think that's quite interesting if you think that these tumors have not had a lot of time to interact with their microenvironment at the early stages. So apparently this phenotype is not just fixed from a genetic perspective, as we've heard from Trevor, but there might also be a similar fixed composition of the microenvironment that can help us to predict the biology over the course of disease. So taken together for this first section, tissue composition analysis with DNN allows analytical robustness, automatization, and standardization, and can provide us with a very high reproducible measure at single cell resolution. I've shown you that these purity estimates are more accurate than visual review or deconvolution, and they can help us to better interpret molecular data. Also, initial data can show that these tissue-based metrics could lead to the development of new and potentially prognostic biomarkers in clinical trials. So this was an example of using prior knowledge and using an AI-guided method to derive an approach of augmented intelligence, right? We know what we're looking for, but we've got a better way of doing it using image analysis. So now let's look at a research workflow where we're really interrogating biology of a colorectal cancer data set in an unbiased way using deep neural networks. So we're trying to predict the molecular features of the disease directly from the H&E image here. I don't think I need to delve deeply into the stratification of colorectal cancer. And of course, we're very well aware that there are deficiencies in our present approach using just morphological factors, that there's lots of work to be done to really get robust genetic alterations or get robust predictions based on genetics. Gene expression profiling can really be a way to provide additional integrative information here with clear biological interpretation that could help us to better inform treatment decisions, because we're looking here at both tumor and host-related factors that might form the basis for clinical stratification. The CMS subtypes have been briefly mentioned. Just to recapitulate, CMS1 is the subtype where pathologists often think about when we see an inflamed phenotype. There is a high frequency of MSI, SIMP-high cases here, and while in early stages we have a favorable outcome, in advanced stages there is worse survival after relapse. CMS2, the canonical subtype, characterized by Wnt and MYC activation, CMS3 by metabolic dysregulation, and CMS4 cases by stromal infiltration, TGF-beta activation, and clinically a worse relapse-free and overall survival. So as pathologists, we've got some indications of what these tumors might look like. As I said, CMS1, an inflamed subtype, CMS4, enriched for desmoplastic stroma. But for CMS2 and CMS3, we really don't know what these cases look like. So we're very limited in actually providing information to the molecular pathologist for testing because we have no prior knowledge of what these phenotypes might present as. So one way, of course, to derive gene expression profiling based information is direct sequencing without prior selection. The other way, and this is what we're very interested in, is to use a deep neural network based classification to make this information accessible directly from the H&E image without need for further sequencing. And the hope would be that this could be a cost-effective approach to both interrogate retrospective cohorts and to look at correlations of these genotypes in a cost-effective manner with treatment outcomes and biology, but also maybe in the future and after further development and validation to use the H&E image as a way to prioritize patients for molecular testing, but also to achieve better clinical stratification for treatment decisions in difficult clinical situations. So for example, in stage 1, could this possibly help to identify patients with adverse outcome or favorable outcome for stratification of surgical resection after diagnosis of a malignant polyp, or in stage 2, to make the decision whether to give adjuvant chemotherapy or not in critical clinical situations. So this study is based on a very close collaboration, of course, with the ESCORT consortium led by Tim Horn and with the Department of Biomedical Engineering, the group of Jens Richter and Koelzer Kunavatana, who's a very talented postdoc in that group. And what we've done here is we've used 3 different cohorts. So the focus cohort with 362 patients over 600 slides as the training cohort, TCGA as a validation cohort, and then the Grampian data set with 170 patients with digital slides and molecular data available in the biopsy setting. So we're looking at 2 very distinct clinical applications, full tissue sections from colorectal cancer resection specimens and biopsies in the preoperative settings. And this is very interesting because oftentimes these biopsies are very, very difficult to profile using genomic methods because of limited material that is available. Here you can see the distribution of the CMS subtypes. As I said, we have all the expression data available to make these calls on these data sets. And as you will also see, there's a significant number here between 24 and 37% of cases where with conventional molecular methods you cannot make a consistent CMS call at this moment. So these in particular, of course, are interesting to interrogate using image-based methods. So how is this study set up? And I'll refer back to the slide that I've shown in the beginning. We've used the sections that were precisely annotated for the tumor regions that were then used for sequencing and gene profiling. Each of these sections was tiled into these small regions and was associated with the ground truth molecular data that we had for this cohort. We used 2 different training approaches. So one training approach to make a predictor that would associate image features with the precise molecular phenotype, and another, which is called domain adversarial training, which was used to reduce the classification weights of features that were cohort specific. So we're trying to reduce the noise within the training data set using 2 different AI training approaches at the same time. For each of these tiles, a prediction was made, and we used a 5-fold cross-validation to basically have 5 different models that would make a prediction very much like an ensemble of expert opinions to achieve final classification. The deep neural network architecture that was underlying this was the Google Inception V3 network, which is a very modern deep neural network, but I won't go into detail of the architecture here. If that's of interest to you, I'll be very happy to address that over lunch. So now, once we've built a trained network from the focus trial, we're now able to make an inference call on unseen samples. Again, tiling is performed, and then for each tile, a call is made based on the image information alone, and the classifier as it's been trained on the training data set. And now this gives us two very interesting bits of information. If you think about how molecular profiling is performed normally, the entire tumor is just scratched off the slide, put into a test tube, and we're just looking at the overall profile, right? So we're mixing everything. We're mixing tumor, microenvironment. We've got no clue where the signal is actually coming from. With image-based methods, you can now use methods of interpretable AI, so so-called class activation maps, to ask two questions. One, is the tumor homogeneous or heterogeneous? And you can get a very, very precise localization of where a certain signal is coming from. And two, what is the ultimate classification of the case? So you reach much more granularity to your molecular prediction than was possible before. And you can do this in a human interpretable way, right? You can go back to these overlay images and look at the regions of the slide that impact the prediction the strongest. So as you can see here in the focus cohort, this shows that there's actually quite pronounced differences within the CMS phenotypes themselves. So there are cases that have very homogeneous and strong calls for one CMS subtype based on the image, but there's also considerable heterogeneity that we've so far not been able to interrogate in a spatially resolved manner. Or if you were planning to do so, you would need to spend a lot of money on sequencing and laser capture microdissection to achieve the same resolution here with the tissue that's under investigation. How accurate is the method? You see that's here for the training cohort, so we're achieving a AUC of 0.88 on the full tissue sections on the focus trial. This is the training cohort, and you see similar performances on the validation cohort, and interestingly also on the preoperative biopsy. So we can achieve an image-based prediction both in the setting of a resection specimen as well as on the biopsy specimen here. Now I've highlighted the samples that were unclassified in the cohort. So unclassified means that we had divergent calls depending on the bioinformatic method that we used. So we used the random forest method that was initially published by the Guinea group, as well as a single sample predictor. And when these calls diverged, we classified these samples as unclassifiable based on the genomic information. An alternative was failure because of insufficient genomic material in small samples. And you can see that using the image, we can now reclassify these cases and resolve them into the different CMS groups in all cohorts. And this is, of course, very interesting also for the preoperative setting, where you can now derive a predictor of gene expression classifiers based on the very, very small preoperative samples. So how do these predictions compare to the baseline associations with secondary molecular features from samples that were successfully classified by molecular profiling? So the black bars here show samples that were successfully profiled using CMS, so the molecular sequencing, and the gray bars show the distribution of the molecular profiles from those cases that were reclassified based on the image. And you can see that there is very little difference between these secondary features, except for the CMS1 class here in TCGA, which is explained by an underrepresentation of MSI cases, which usually were correctly classified by the CMS, so the molecular subtyping in the beginning, and were therefore underrepresented in the image-based cohort. So what is very interesting to me as a pathologist now is that we can go back and now learn from the AI-based prediction what these different CMS subtypes look like. And if I refer you back to the slide that I showed you at the beginning, we know that CMS1 cases are often mucinous. They have lots of infiltrating lymphocytes. And you can see these features coming up in the CMS1 tiles here that we've identified. The same is true for CMS4 with lots of stroma, tumor budding, signs of EMT. But now, interestingly, you can also get an idea of what the CMS2 and CMS3 subtypes look like, with CMS2 showing this cribriform pattern and comedo-like necrosis here, and CMS3 with a papillary architecture and lots of mucin production. So we can actually now begin to learn from the AI what the impact of certain genetic alterations may be on the tissue level, and close that loop again to better understand the biology downstream of this. If you look at high resolution, again, these are the class activation maps showing those regions that were most informative of the prediction. Again, you can see that in CMS1 and CMS4, both the epithelial and stromal components were informative, while in CMS2 and CMS3, the epithelium was most important for the call. Looking at heterogeneity, I think this is a very, very interesting result. We can resolve heterogeneity on an image level now, but is this correct? So what we did is we derived a secondary call based on the RNA sequencing data by relaxing the threshold for the random forest classifier. And you can see that the calls made for CMS in a quantitative way between the image and the RNA-seq correspond largely. So we can now use the image to interrogate heterogeneity, or get a suggestion of heterogeneity on the slide, just as with a molecular method, but we can do this in a spatially resolved manner, which of course opens avenues for downstream investigation of what actually is driving changes in these areas, be it microenvironment features or changes in tumor genomics itself. Last, of course, we're very interested in prognostic stratification. So this is the stratification of the focus in the TCGA datasets based on molecular CMS. And you can see the expected correlations with CMS1 doing poorly here in advanced disease, and a split here between the CMS groups as expected for overall progression-free survival. Now, if we use the image-based classifier, you can see that we can largely reproduce these associations, particularly in the focus group. And interestingly, now if we use this predictor on TCGA, for example, you see that we're also pulling out the poor prognosis CMS1 subtypes here in particular, and we're presently further investigating the specific features here that are driving this image-based prediction. So I've shown you some research data now that of course needs further validation and exploration, that we can use a computational model to predict transcriptional subtypes of colorectal cancer from standard H&E sections. We can use this approach to now make sequencing information interpretable, because we can closer associate the morphology, molecular features, and outcome data. We can use these approaches, be it CMS or others, to classify samples that were previously unclassifiable by RNA expression profiling, which can give us a novel insight into tumor heterogeneity and gives us access to novel prognostic information. And last, we can enable stratification of patients in diverse clinical settings, because we can apply this method to any type of specimen, be it a preoperative biopsy or a resection specimen. So this really opens a new way to interrogate both prospective cohorts in clinical trials in a very efficient and hopefully inexpensive manner. But we can also achieve retrospective interrogation of existing data sets that would otherwise be unable to be sequenced just because of the prohibitive costs. So we're now collaborating with the SCOT trial group to perform image-based molecular classification across the over 3,000 cases of the SCOT trial, which is an international randomized phase 3 trial done at 244 centers, comparing the efficacy of 3 versus 6 months adjuvant oxaliplatin, a 5-FU-based therapy. And we're very keen to look at the correlations with chemotherapy outcome in the light of recent studies that show potential value of CMS classification to making a predictive stratifier. What's next? Of course, I've alluded to the potential of interrogating the specific areas where we get an image-based call, and to better understand what is going on here in terms of tumor microenvironment interaction. And digital pathology, of course, has many, many different levels. We can use this now to add spatial information to the molecular data, and this is where we're going. We're using these multiplexed immunofluorescent approaches to localize the different compartments, so both the tumor cell as well as immune cell compartments in a highly multiplexed way in collaboration with Oxford. And this now allows us to assign a spatial location to each cell type and to then derive downstream metrics, for example, nearest neighbor analysis between each of the different cell type combinations and proximity analysis, which we can then correlate with the molecular phenotypes to better understand tumor microenvironment interaction. And this brings me back to my last slide. I think I've highlighted a number of orthogonal approaches where we can use the image to look at both features driven by prior knowledge, but also the context of morphology and molecular classifications in a new way, and sometimes in an unbiased way. And we can begin to learn from these predictors made by AI to better understand the biology of disease. So with that, I'd like to thank all the people that were involved, in particular, of course, Tumour and everyone involved in ESCOT, and the group of Professor Jens Richter at the Department of Engineering Science in Oxford. This has been a very productive collaboration and very, very positive in moving this forward. And I'd like to in particular also thank all the collaborators here that were involved in the clinical trials that have made the future collaboration possible here on the SCOT Trials group. And with that, I'd like to thank you for your attention. I'll be happy to take any questions.